happening now is we don't talk about the pandemic being on now. The pandemic has ended. Do we still see cases? Yes, but we don't see as many because through both being exposed and having had the disease and through having vaccination, between everything, more and more of the population is resistant, and so therefore fewer cases. And so that's kind of where we're at, and then that's where it starts to become always present, kind of like influenza. Are we still going to give you boosters because it keeps changing? Probably, unless we can come up with that, new vaccine that always works. I love this. You can actually look, if you want to see a report card, you can actually look at America. Okay, so it's been a little while from the data, but if you look at the individual states, you'll see the darker the color, the higher the vaccination rate. The lighter the color, well, white means they don't have the data. So can you see, like Alaska there? We have no data for that. But amongst the states that have data, who's the darkest one? Well, it turns out ND. ND stands for North Dakota. So isn't it interesting? North Dakota parents love their children the most. And who loves their children the least?
Nevada.
Where are we? There we are.
Yikes.
We're not that far behind compared to Texas.
We're not great. But you're like, oh my God, even Texas is better. And we never say that, do we? Do you know they're better with homeless population as well? Do you know they have more facilities for homeless? Do you know why? No. It's because there's better communication. Oh my God, is that my theme today? There's better communication between all the agencies. So you know how there's like city, county, state, health, political, mayor, not-for-profit groups, they all work together. They all work together. Who knew? We get kind of a D-.
I especially didn't hate women.
Oh, well, that's another area.
Because they're gray, I guess. And every other sense they're gray.
Yeah. Oh, you mean Texas?
Yeah.
Oh, okay. All right. Well, I'm going to move on from that because that's beyond my purview. All right. So herd immunity. What percentage do we need? Now, I want you to notice, can you see something called an RO value? They always quote that, and that is to do with how many people can you infect. Remember, we talked about pathogenicity. If there's you in an elevator, right, with 10 other people and you're a sicko, if it is very pathogenic, you'll infect 10 people. But if it's a lower pathogenicity, meaning less infectious, you may only hit one person out of the 10. So check it out. Can you see if you have like polio, you can spread it to 5 to 7 people. Check out which are the most infectious of all. You see measles, that would mean you'd hit everybody in the elevator, plus the next 3 to 5 people getting on the elevator, and then the 3 people on the next floor after that. In other words, very, very communicable. And can you see whooping cough, still, which is why they're pushing shots for grandparents now, because they're like, you don't want to kill your grandchildren, they're babies. So what's interesting is if you look at the percentage needed to prevent a public health issue, so in other words, we get good or herd immunity, you know what that means now. Can you see that the more infectious it is, the higher number we need to vaccinate? That makes sense, doesn't it? We want to make sure we protect as many people as we can if it's so easy to communicate. So there are different types of immunity, and I want you to notice, can you see with immunity, you can have what we call passive immunity. What does passive immunity mean? It means not you. You know how we always talk about making B cells, right? And making antibodies? If we're talking passive, you didn't make them, something or somebody else did. So passive means what? You receive them. Okay, so now then, you can receive antibodies naturally. How? Okay, during pregnancy, this is what we were just talking about, right? During pregnancy, Remember, through vertical. Remember, the IgGs go through that way, and then the IgRs if you breastfeed. Is breastfeeding natural? Again, some people are weird about it, but yes, it is a natural part of being a human being. No labs involved, right? No labs, no needles, just natural. Very good. So can you get antibodies? You can when you're a fetus and when you're a baby. And did you make them? No. Who made them? Your mom made them, right? Is that natural? Yes. So can you see the term passive? And then can you see the word natural? Are we good with that? All right, can you get artificial? Yes. That means passive, you did not make them, but you received them from somebody else. But is that normal in a human life? No. Usually the only way you get antibodies is you make them yourself. So who made them? Well, they could be made in a laboratory. They could even be made, can you see my horse here? I know it looks like the knight in shining armor. It's the horse that's the important bit here. Why a horse, you might be going? Well, you can get antibodies from another human being, but you've got to what? Extract them, pay the person, purify them, make them safe, and then you've got to do the IV. Not a cheap process. A horse is a mammal like us, warm-blooded mammal, makes antibodies like us, and it's big. So guess what? You can have them make lots of antibodies fairly cheaply. And then you can get the antibodies. Now, would that be passive if you get them from a horse? Yes, because who made them? The horse. Did you have anything to do with it? Okay. And then you got them because what? You had to go to the clinic and then have an IV or an injection of some kind, right? That is not normal, natural human life. Okay. It's a good thing, and it saves lives. So do you see what we talk about, the two terms? Passive, meaning you didn't do it, the horse did, or maybe another human, but it's artificial. Why? Because it's either through laboratory technology. Do you understand? Technology meaning injections or IV. Okay. So do you see how we use the two terms? So that would be what we call passive. Now let's talk about active. So who makes the antibodies? One word. You. Or you could say me, right? Either one will work. You make them. Can you think of a natural situation where you would naturally be making antibodies and memory cells? You're a sicko. You're a sicko. Do you get sick as a human? Are you living on a planet where there are pathogens? So that would be what? Active, meaning you did it, you saved yourself, and it was through natural exposure. Life is, well, dirty. Okay, so the two terms are what? Active, natural. Okay, now let's talk about the other kind of natural, the other type of active, where again, who makes the antibodies? You. But it's not through normal life. Not through normal life. Not through just being a human being, eating, drinking, pooping, having intimacy, whatever. Okay? Artificial. Again, what's artificial? That means we're using technology. So that would be like your flu shot, or your SARS shot, or your DTaP, or your MMR, right? So I have a page for you to play with at home where you can play the game of which terms apply. Are these ones, right? The next slide. You see all the photographs? Don't look at the answers, but play the game at home and see, test yourself to make sure you understand it. Yay! Are there diseases you can get from breastfeeding? From the surface of the skin, but then they've been interacting with them, and they're actually acquiring their microbiota. If they're healthy, then they should be protected by mom's antibodies. And so even if there were something from the breast milk, that should protect them. I think I'd have to look it up, and I'm sure if someone's got a smartphone, Can you plug it? Anyone got a smartphone there? Can you plug it in for us? Eric and I want to know. Breastfeeding, transmission of pathogens. Try those keywords and look it up while we're still talking. It's interesting. I'm just thinking in terms of, with all the benefits of breastfeeding, I could see if you had lesions there that was oozing pus, that might cause from there to the mouth. Could you see that? Yeah.
Say if another has breast cancer and doesn't know, would that affect anything?
If the mum
Has breast cancer and doesn't know.
breast cancer won't affect the baby. Yeah. I mean, they might not allow it. Go ahead.
So it doesn't say specific pathogens, but it says it can occur through direct contact with body fluids, droplets, or airborne spread.
Okay, so it's direct body contact. It's saying the transmission routes, but it doesn't give a specific example, so I don't believe it. I mean, I believe that transmission occurs, but we're looking for what infection, don't we? Maybe we have to do a slightly different search. You just keep looking. Maybe you have to say diseases.
It says bloodborne pathogens in breast milk may include, but are not limited to HIV, Hep B, Hep C, West Nile virus.
So it does sound like more like certain viruses, doesn't it? Now, hepatitis B, remember that one's bloodborne? You could get that from sharing a toothbrush, or even if there was blood in the saliva, direct contact. Do I think it's that common? No. And the fact that we're really having to hunt it down kind of tells us that.
I did find that the CDC just recommends any potential pathogen in breast milk to be treated as like an accidental exposure to body fluid, because the actual transmission of the agent during ingestion is really low.
So low numbers. Yeah. I think it's we have breastfed for millennia, and they definitely, babies are healthier with. Again, it's risk assessment, I guess, isn't it?
And I've donated breast milk and also have used donated breast milk.
Yes, donations of breast milk occur. So yeah, that's true. So I think it's one of if you're going to exchange a body fluid, that's one of the better. Is our conclusion. Okay. Very good. I think that was very interesting. Good job, everyone. And the point of the fact of the matter is, if you've got a disease like that, how likely are you going to express milk? Right? If your body is suffering, you probably, one of the first things to go is you stop, if you're sick. Okie dokie then. And you can always express it. And then so that there isn't that body-to-body contact. So if you had, like you were saying, direct contact, and droplets, so if you didn't want to directly feed the baby, you would express your milk using one of those lovely pumping machines, like a cow, and then you can freeze it, freezes really well, and then you can have the baby have it without having direct contact. So I think that's interesting. Okay, let's talk about, this is a pivot slide, everybody. This is the one that, from this slide, you should be able to teach the next one of slides. So we're going to go through vaccine types, and we'll talk a little bit about how you can give antibodies when they're a sicko, and give them an instant titer, which is like a miracle. And then we should talk about the newer vaccines. So my first one that you need to know is that a whole cell alive vaccine has a specific name. Circle it, it's called attenuated. Everyone say attenuated. Attenuated, that means it's alive. So it's alive. Now, it has been treated so that it is weakened. It has been treated so it's slightly altered. And we say it doesn't cause disease. Can you see the term we use? Virulent. But it's still alive. And so what's amazing about it is it acts like the real thing without causing disease. So what does that mean? It does the A word. It does the colonization, where it's doing the, dividing. And so you get lots and lots being produced, and that really wakes up your immune system, because it's like the real thing without having the actual disease. Okay. Okay, so that is the most likely real thing. So you'll have the strongest memory T and B cells from that type of vaccine. What's the downside? There's always a downside. If you are immunocompromised, then you are weak, aren't you? And you have no defenses. And the problem with a virulent form is perhaps it might have enough mutations that it might revert to being the real thing. Unlikely, but always possible. So do you want to take the risk? And the answer is no. Would you want to give it to a little human that doesn't have a fully developed immune system yet? Because again, if you don't have a proper immunity, that might be a little bit too much of a risk. Keeping them alive means you've got to store them in very special conditions, which isn't always practical. But on the good side, strong, long-lasting. So you have one shot, you're probably good for life. Never have to have boosters. You see the advantages. But you see, it's a double-edged sword, everyone. Double-edged sword. Okay, what's the next best? The next best, are the ones that are dead. But I forgot to give you examples. I'm so sorry. Here we go. You need to know your examples. MMR. Now, I know it just says measles. Know that that is red measles, the bad one. Mumps, which is not on our disease list, but can cause issues. And then rubella is the other measles, German or 3-day measles. Tuberculosis, you do not get that in this country. Some of us did, because we're from another country. There's something called, there's a polio one that is live called the saving polio. You as an American did not get the live one because we don't have it. Polio is virtually non-existent in this country, except for people coming in occasionally through immigration or through travel. So it is incredibly rare. So we give this, the live one, which is a little bit riskier, we give it still to children in countries where it's a problem. In other words, where they're, poor sanitary conditions, more likely to have, the really, really bad version of the disease. Heard of varicella? Varicella is another name for chicken pox. That's right. So those are examples. So this is one polio. On the next slide, you'll see the one that we do get, okay? Which is the much safer whole cell but dead. Okay, if you're dead, you cannot cause an infection. But do you have epitopes and antigens? And if you're a whole cell, you're going to have lots and lots, aren't you, that should get your immune system going. So in micro-speak, if you're dead, we don't say you're dead, we say you are inactivated. So you have to use the correct term, so circle, inactivated. So it's dead, and obviously it can't reproduce. I know you should know that, but we put it in there. But you always have people going, oh well, it gave me this such and such disease. And I'll be like, that's a miracle. It's dead. If it's dead, it can't do anything. But remember that when you get those antigen-presenting cells, remember that kicks off your immune response, and it doesn't always feel good. You remember you get a bit of fever sometimes, and you get the aches. So it's dead, which means it's safe, everybody. It's safe. And examples of the safe. Notice there's a polio that's the safe one. This one's called the Salk polio. Sorry, Salk. S-A-L-K. So the other one's the saving, and this one, the dead one, is the Salk. That's the one we get in this country. Now I had the more dangerous live one because I'm old. But they make everything safer these days. Hope you never have to have a rabies one. I don't know why I had it twice. Horrible shot in the stomach. Horrible. But it's dead. The flu shot, influenza, as you know, is dead. That's the commonest version. Hep A, if you get it, is dead. Cross out the other rabies. Yeah.
What I'm curious about is, like, for example, with the rabies shot, people tend to get it when they've been possibly exposed to rabies. But what I don't understand is if you've already been exposed to it, like with a bite, and how would the shot have you acted with that?
Then you have to understand about the pathogen. How come a shot How long does it take for a shot to work? This is a great question, so let's think about it. You can figure this out, because you're smart. Okay, so the microbe, right, gets in through the bite, let's just assume, okay, and you get the vaccine the same time, or within so many, a day, whatever, right? So, how long does the vaccine take to work? Do you remember? Tell her, she can't remember, she's like panicking. Can anybody remember and help her? Oh, okay, so can we give them some? You do know that a vaccine doesn't work for 10 days.
Oh.
We could go through that. Well, you remember that bit, don't you? But can you apply it? No! Okay. 10. I'm in a bit of a bend. 10 to 14 days. Now, if the vaccine works, obviously they wouldn't bother giving it to you. What do you think is going on with the pathogen then? Uh-huh.
It's in low numbers.
Uh-huh, and? There's not a lot, and? Mitosis, so, or binary fission in this case is slow. Okay. Told you could answer it yourself. This is what I'm training you to do now. It's like, try and think your way out of a paper bag. Even if you don't know the answer, you might. And a lot of you know a lot more than you think. And that's something you've really got to bear in mind. You know, people have low self-esteem. Okay, next on the list. I'm not leaving anybody out. I can whack them. The next two slides are similar. They're both parts of the pathogen. So you can see not as many stimuli for your immune system. And so we actually divide them up into, I call them the bits and bobs, but for goodness sake, don't call it that. We call it subunit, which means bits. And then the other one is the bit that's the toxin. You know, the toxin is just one bit of what's produced by the pathogen, but we separate them because toxins are so important. All right, so bits and bobs, subunit. What could we pick on? Anything, a bit of the flagella, maybe even the polysaccharide coat. But the problem is when you only choose a small piece, it may not be big enough. Do you remember, to be an antigen, usually it has to be what? Big, and protein is better. Well, for some vaccines, we actually use, you know, the attachment, not the spike this time, but the capsule. You know, some of those microchip capsules, that's made of polysaccharide. And so we want to get the antigenic portion, so this little piece, remember, a bit, portion of the pathogen, and that could, you know, and if possible we choose protein, remember? But what's the next best thing? We might have to use like a bit of carb in the capsule, for instance. Now the good thing about this, as I said, the good part is it's safe, because it's definitely, it's not even a whole organism that's dead, it's a bit. And so we call it, if it doesn't have the cell, it's just a piece of the cell, we call it without cells, or acellular. Now the problem is that your immune system doesn't see it very well, so we make it big, because microbiologists are good at that. And what they do is, just like when we talked about haptens, remember how something can become big if it attaches to a carrier protein. And so we can do that in the lab, and we call it a conjugate. So conjugate, everyone say conjugate? Conjugate. Very good. And so that means we can take a little subunit, like a bit of a capsule, and we complex it, ideally with a nice big protein. And we call it a protein carrier. And that means, look at it, it's going to be big, and it's going to have protein. And our immune system's going to go, whoa, okay, here we go. So we do that. There's a vaccine called DTaP, D, T, and do you see the acellular pertussis now? AP. Okay? We'll get to the DTs in a minute. Sounds a bit weird, doesn't it? Okay, so we've got whooping cough would be a good example. Again, these all kill babies. The influenza, that's not a flu. Lots of things are called flu, but this one is brain damage. Meningococcal meningitis, babies, brain damage. Pneumococcal pneumonia, babies, but now we've got like respiratory system. Yeah? So you see, very useful when you want to get something really, really safe to a baby that could die of it. The problem is you might have to give a bunch of them. So what do you call a bunch of them? Remember the B word? Boosters. Because, and you have to make sure you get them all. So why is our vaccine protection low in the United States? Remember I said in the 70s? It's because, not because they don't get the original shots, it's because they don't do the follow-up. And research has shown that if you don't get your boosters, you don't have the protection that you need to be safe. Okay, so the other bit of the subunit is very specific. We're not going to give you the toxin because that would kill you. So remember, we call a modified toxin a toxoid. Modified toxin. Is it safe? Yes, we made it safe. And remember, that stimulates a special class of antibodies. Remember there's different classes? This one's called neutralizing, because it stops the toxin getting to its target. So it neutralizes. And there's another name for it. Remember the word antitoxin? Same thing. Okay, here's the DT, everyone. In the shot, DTAP, DTAP. The D goes against the horrible diphtheria toxin that kills babies, and the tetanus toxin that kills everybody if you don't get it. And that's another one. We wait, don't we, until you've had a cat as an adult, not as a baby now, because you have your series. But when you're an adult, you get your foot or you piece of glass or something. You're like, when was the last time you had a tetanus shot? And they're like, right, get your ass in here so we can put a needle in your ass and get you out. It's the same time. So again, we've got to think about the biology of it. Yeah? You said right. Get the hang of this. Recombinant means we're going to make things safer. Sometimes one little epitope isn't enough, and we need to have lots and lots of copies, and we want to make them really, really quickly, cleanly, and safely. So we use a microbe as a factory. You know, you've heard of yeast, haven't you? Did you know that yeast is the most marvelous factory of proteins? And you can take a gene from a pathogen, pop it into a little yeast cell, or a billion of them, and they will happily churn out the products. And that would be little antigens and epitopes of a different species. So do you see the word recombinant? Recombinant. What does that mean? It means we're combining genes. So what are we combining? Genes from a pathogen with the genes in our little fungus, our little yeast factory. And so we can get lots and lots of copies. So that's what it says. A gene of the pathogen is placed inside another organism, such as a yeast. What's the product? Remember, we call it a recombinant. And it then, when it divides, you get many, many clones, and it produces many, many, many copies of that epitope antigen. And so, anyone had their Hep B series? That's how it was made. It was made by a yeast, and that you can just take the nanoparticles and get it injected. So it's cheap, fast, and safe. And it's a subunit. It wasn't the whole thing, it was just a piece of the Hep B. Now, you know, influenza, they actually take multiple variants, because you know there's more than one every year, influenza variant, and they take bits of the spike, and then what they do is they take a piece from this variant, a piece from that one, and a piece from that one, and so you end up with this wonderful recombinant vaccine that has a bit of this, a bit of that, and a bit of the other from different strains, so you get a better kind of protective effect. So what are examples? Hep E, hepatitis B, human papillomavirus. Remember that's the one where it prevents papilloma warts. Remember they call genital warts. And then, of course, influenza. Yeah, so safe and clean. If you are a sicko, for example, you've been bitten by a snake, you don't have much time. So what will we do? We'll give you the antitoxin, which will be antibodies that we've made somewhere else. Is that artificial? Yes. Is it passive? Because yes. And so we take antibodies that are specific to whatever is the problem, whether it be a toxin or pathogen or whatever. what happens to your titer? It's instant. Love it. Instant protection. Instant health titer. Yay. Now, you've got the antibodies from somebody else, right? You're not making them. Will they last forever? So therefore, did you know that proteins usually last just for a couple of months? 2 to 3 months in general in your body? And so it's temporary. And where can we get them from? Anybody who makes antibodies. So we could get them from human survivors. But that's very expensive to get a whole bunch of humans together to get enough antibodies. So what's the cheaper method? Use a horse. And so we use, horse. Yeah, I know. Okay, well. Okay. Has any horse get harmed in the making of them? Not generally. So that's good. I mean, they don't slaughter the horses, which they do for their meat and whatever. It's like the other things. All right. So what I want you to notice, France, that eating horse flesh, right? Certain countries, cultures. It's Mongolia originally, historically, kept the whole cultures alive, horse meat. Are you going to eat a horse in America? Perhaps not. You never know if you're starving. Okay. All right, I want you to notice the end. You know, I'm always training you to read the word, the whole word. These are drugs that are given to you when you're sick of things like cancer, autoimmune diseases, and they're often advertised on TV. They'll say, take this and win the drug. And it gives you the proprietary name, but if you look at the active ingredient, it always ends in MAB. Do you see MAB? Check it out. MAB means antibodies. Monoclonal, meaning, remember we want something dead specific, monoclonal antibodies. There are drugs now that treat breast cancer. If you brought up breast cancer today, there are drugs that treat leukemia and melanomas. They're heavily effective. City of Hope has a whole translational protein department where they definitely look at options. Other ones for asthma and so on, rheumatoid arthritis. And I don't know if you remember, but for the bronchiolitis, remember little baby bronchiolitis, RSV bronchiolitis? Synagis was one drug. It actually ends in MAB when you look at the active ingredient. Okay, so let's talk COVID-19. One way to make you immune is to give you a little piece of DNA from the virus and actually get it into your cells, and then your cells start expressing, can you see viral proteins? Can you see? Now how the heck do you get the DNA in from that SARS virus? Use a vector. Can you see my little friendly little virus vector? This is a virus that's not going to give you disease, but it's very good at doing what? Injecting DNA. You know how viruses can inject? So the vector injects it, you express it, it gets presented, remember, to the antigen presenting cells, and then you get an immune response as to it. And so yeah, DNA viruses were produced, and if you use DNA, it's much more stable than RNA. Okay, but I don't think we got those. That was more AstraZeneca. A few people, some people did in America, but that was more the European. So what are we coding? What is the DNA coding, the spike protein? Where's it carried? It's inside, remember I told you the little vector? So inside, it's a type of virus called an adenovirus, A-D-E-N-O, an adenovirus. And it has a tough protein coat, which means that our body will leave it alone until it's delivered. Because remember, to be a good vector, you've got to successfully deliver package. Works very well. And your cells express it, but only for a short time, because the DNA degrades, and also the protein, the proteins degrade, and and so on. So it can't, the virus has no effect, but the DNA is transcribed, translated by you into little spike proteins, which then represent. Now the RNA ones work similarly, but it's one step closer. But before I get to RNA ones, which is the one we had, did you know we could use a different vector? So in this case, we've used what? A virus to do the delivery. We could also use a small circular piece of DNA that we can inject to you through the plasmid. Remember plasmids? It's just a very, and that can have the spike gene in it. So, you know, so this is, you know, they're very, what's the really cool thing about these style of vaccines is you can do it really quickly and easily. So you know when we had the COVID-19 issue and it took, I think it took them a month and they sequenced it, which is amazing. They actually managed to make a vaccine within 2 weeks. But it took 6 months for it to be approved. And so that's why it didn't get onto the market. But it's once it's approved, you can literally give a whole population to get it going in a week. That's how powerful it is. People said that these DNA vaccines are new and we don't trust them. Sorry, we're coming to the end of our time here. But they're new and we don't trust them. Rubbish. We've been using these type of vaccines for 10 years before we even did it for COVID. So in this case it says we use, what did we say, a piece of our little pathogen, our virus, goes where? In a plasmid, injected into your skin or muscle. And who makes the spikes? We do, it gets presented quicker. RNA is faster because you don't have to do a transcription because we've already got RNA, right? So now the RNA goes in, but this is even more elegant. We don't need a virus, we don't need a plasmid, we just coat it in fat. And fat merges with our plasma membranes and of our cells, and so it gets delivered. And that was the hardest thing. What's the word for fat?
Lipid.
Lipid. That was the hardest thing to develop, everybody. It wasn't the vaccine, it was getting it coated so that it wouldn't be destroyed before it could make the delivery into our muscle skin cells. Okay, very good. We'll finish this off on Thursday, but it's basically, I just think